Hello, everyone. My name is Sean Hoban. I work at the Morton Arboretum, which is outside Chicago in the United States, and I'll be giving just a short introduction to the Genetic Composition Working Group to help transition us into the genetic composition session of talks for today. There are three co-leads to the Genetic Composition Working Group: myself, Anna MacDonald at the Australian National University, who's working on the Oz Mammals Genomics Initiative, and Maggie Hunter, who works for the United States Geological Survey. We also have a steering committee, which helps direct the working group and design some of our structure and initiatives. The steering committee is Libby Liggins, Martin Breed, Jessica Da Silva, Joachim Mergier, Rodolfo Jaffe, David O'Brien, and Laura Bertola. The mission of our working group is to provide the necessary data, tools, and knowledge, especially tools, to better monitor and safeguard genetic diversity within all types of species, so wild, managed, and cultivated, ultimately to benefit human well-being and ecosystem resilience, so society and nature. To achieve this mission, we focus on making data usable for scientists, so improving tools, models, methods, databases, standards, repositories for data, metadata, etc. We also work to make genetic data, tools, and genetic monitoring accessible and translatable for policy. So making it visualizable, helping with interpretation, developing simple but effective metrics and indicators. Our focus is within species, as genetic diversity has been defined by the Convention on Biological Diversity. The variation within species that helps them adapt. It's the foundation of other levels of biodiversity. Ultimately, it results in species diversity. It also helps make ecosystems more resilient. It's the source of adaptation. Genetic diversity is also protected. Again, it's one of the main targets of the CBD. It's in one of the main goals of the Zero Draft for post-2020. And genetic diversity is recognized and protected and monitored under numerous national legislations as well. Our working group has well over 100 members, growing all the time. About half of these have been active in one of our projects, some of which you'll hear about today. We have a large number of countries represented on the inhabited continents. We have a high emphasis on diversity for our group, and we represent different ecosystems as well. So a few of our projects, most of which you'll hear about today. One of our main initiatives has been to define genetic composition EBVs, the state of the field, what are the workflows, what happens to genetic data, how it can be reused, models for genetic data, and policy applications for these genetic composition EBVs. After these are defined, another project is simulating how these EBVs will respond to population changes. We've also written a comment article on the CBD Zero Draft released earlier this year, proposing changes to the goal, an action target, and feasible indicators connected to genetic diversity, as well as a short critique of current CBD indicators and future work in genetic monitoring. We also have a project which you will hear about in the policy session, a review of genetic diversity considerations in the CBD national reports over the past 2 reporting periods. And a major element of all our projects is policy explanation. So we have developed some policy briefs to connect our projects, summarize and connect our projects to policymakers. This is part of one of our policy briefs. It's a 2-page attractive, accessible summary of our commentary article on the CBD goal, action target, and indicators for genetic diversity post-2020, which has been accepted at Biological Conservation and should be out any day now. If you'd like the full policy brief or the paper, just let one of us know. To prepare you for the rest of the session, just to let you know, we've mostly resolved our 4 genetic composition EBVs. So genetic diversity, long recognized as an important part of genetic composition, has 2 elements, richness and evenness. Genetic differentiation, both how many genetic or population units and how different they are from each other. Inbreeding, and effective population size. These together determine a species' ability to adapt and respond to environmental change. So I hope you find great value in the sessions today. A couple things you can do. Please continue to attend these sessions. You can see our poster on the working group. You could also attend the EBV 2020 sessions, which will give an update on EBVs and kind of their future and indicators for them. Please join us for the all hands genetic composition meeting later in the week. Sign up to participate at this web address and join our group if you're interested. Thank you very much.